A very good day, everyone. So, welcome to our YouTube channel. This topic, today's video topic would be apoptosis. So, before getting into the video, those who have not subscribed for my channel, do subscribe. And if you find my video useful, please do like and share. And thank you so much for all the support until now. Okay. Fine. So, first of all, who is the target audience for this topic? All the life science students. Okay, it's just not your CSAR, UGC NET aspirants. All life science students, cell biologists are the target audience. So, again, apoptosis comes under unit cell communication and cell signaling. Okay, unit 4 in under your CSAR, UGC NET. So, what is apoptosis? What is the mechanism of apoptosis? And why is it so very important in our body for apoptosis to take place is all that we are going to study in today's class. Okay, so first of all, apoptosis is defined as energy dependent biochemical mechanism of programmed cell death. Okay, every cell has to undergo death. Okay, every cell has to die and new cell needs to be formed, right? So a cell will die, a cell will age. Okay, aging of cell is called cell senescence, and that cell, once it is aged, it will undergo programmed cell death called apoptosis. Okay, there are two things which we generally get confused with. One is apoptosis, another one is necrosis. Okay, so what is the difference between apoptosis and necrosis? You can see this picture. Okay, apoptosis happens when a cell is injured. Okay, when a cell is infected or when something is really wrong with the cell. Okay, that time your cell will undergo necrosis. Okay, whereas when the cell has attained its maximum cell division, or it has undergone its maximum number of cell division, it will undergo apoptosis, or when the cell is aged. Okay, so apoptosis is a very natural process, and necrosis happens whenever there is a cell injury. Okay, so it's very important for us to understand the difference between apoptosis and necrosis. Both of them lead to cell death. Okay, but how they are leading to cell death is what is very important for us to understand. Okay, so it's a apoptosis is an energy dependent biochemical mechanism. It depends on energy, depends on a lot of genes and everything. Okay, whereas necrosis is an energy independent. It doesn't require energy. Okay, that results from acute tissue injury. Say for example, a person has undergone an accident. Okay, because of accident the person has lost his cells. Okay, so that could be necrosis. I hope it's clear. Apoptosis is cell suicide. Necrosis occurs due to cell injury. Okay, both are cell death. That much must be understood. Okay, so what are the characteristics of apoptotic cell? So whenever you view a group of cells under the microscope, how can you tell that these cells are apoptotic? You can actually tell these certain cells are apoptotic. How can you tell? The cells become more compact. Okay, the cells will become very compact. Okay, they come very close to each other. Blebbing occurs at the membrane. So if this is usually if the membrane is like this, there will be blebbing like this. Okay, so the membrane will start blebbing. Then chromatin becomes more compact and fragmented. So what is chromatin? Chromatin is a mixture of your DNA and protein, DNA and your histone protein, right? So chromatin becomes even more compact and it becomes fragmented. Okay, then chemical change occurs on the surface of the membrane, which helps phagocytes to recognize and engulf them. So the plasma membrane, the membrane here we are talking about is the plasma membrane. So the plasma membrane undergoes certain chemical changes. Why these chemical changes occurs? Because the phagocytes must recognize them and engulf them. Okay, now what are phagocytes? Phagocytes are immune system cells which help in phagocytosis or cell eating. Okay, next, what one such chemical change, okay, one such chemical change is that the phosphatidylserine. What is phosphatidylserine? It is a phospholipid. Okay, phosphatidylserine is a phospholipid which is present in the inner leaflet, flops back to the outer leaflet in apoptotic cell, which marks as a marker. So to put it in a simple word, there are usually two membranes, right? The plasma membrane is a unit membrane. It's composed of the outer membrane and the inner membrane. Okay, so initially in the normal healthy cell, this phosphatidylserine is present in the inner leaflet. Okay, but when the cell is marked for apoptosis, this phosphatidylserine moves to the outer leaflet, and hence now the cell is marked for apoptosis. Okay, now the phagocytes will recognize the phosphatidylserine in the outer leaflet of the plasma membrane, and hence helps in the recognition of the cell for phagocytosis. Okay, I hope it's clear. These are the very important characteristics of apoptotic cells. Okay, we shall move on to the next slide. So this apoptosis depends on something called as caspases. Okay, caspases are nothing but cysteine dependent aspartate specific protease. Okay, so first of all we shall understand what is a protease. Protease is a enzyme which is going to cleave the protein. Okay, I hope everyone knows that. Okay, aspartate specific means what? They are going to cleave the protein at aspartate residue. What is aspartate? Aspartate is an amino acid. Okay, and they depend on cysteine. Okay, so caspase stands for cysteine dependent aspartate specific protease. That's called the caspase. So what do? They cleave protein at the C terminal. So you all know a protein will have a N terminus end, that is your amino terminus end, and your carboxy terminal end. Okay, so they are going to cleave the protein at the C terminus end at the aspartate residue. Okay, now once caspase is activated, apoptosis is irreversible. Okay, once the caspase protein has been activated, the entire apoptosis is irreversible. Okay, and after that, no one can stop the apoptosis process. Okay, fine. So there are two types of caspases, initiators and effectors. So initiators involve caspase. These numbers are very important. They are usually asked in your exams. Okay, caspase 2, 8, 9, 10 are initiator caspase. Okay, whereas your effector caspases are caspase 3, 6 and 7. Okay, these are the effector caspases. Now, inactive procaspase is activated into active caspase via cleavage. So, what is this inactive procaspase and caspase? Say, for example, this is one caspase. Okay, this is called the procaspase, which is in its inactive form. So, now this procaspase will be activated to its active form via cleavage. Okay, it undergoes, the procaspase undergoes cleavage to form active caspase. So which is longer, procaspase or caspase? Procaspase is longer than the caspase. Okay, so what have we understood? Caspases mediate apoptosis. Okay, they are nothing but cysteine mediated aspartate specific protease. They cleave proteins at the C terminal. Okay, there are two types of caspases, initiators and effectors. We will study more about them in the coming slides. So, inactive procaspase is activated into active caspases. Okay, fine. So, initiator caspases activates effector caspase. So, what do? Initiator caspase is going to activate the effector caspase. All this caspase 2, 8, 9, 10 is going to activate your caspase 3, 6, 7. Okay. So, that is why we call them as initiators and effectors. Next. What is the target of this caspase? What do they do? I told you they are proteases. So these proteases are going to go and cleave or going to degrade your nuclear lamins. Okay, your lamin proteins. So what are nuclear lamins? They are the intermediate filaments that are going to be there, that are going to line your nucleus. Okay, your cytoskeletal proteins, your microfilament, microtubules, Golgi matrix component, and hence what they are going to do, they are going to cause a disruption of the entire cytoskeletal structures. So once the cytoskeletal structures are being disrupted, the cell will be marked for death. Okay. So caspase dependent pathways, there are three types of caspase dependent pathways. One is your extrinsic pathway, intrinsic pathway, and perforin pathway. Okay, so extrinsic pathway is like death receptor pathway, intrinsic is mitochondrial, okay, and perforin is granzyme. We shall study each one in detail. So first of all, caspase dependent extrinsic pathway. Okay, so extrinsic means there has to be a signal from outside. Okay, there has to be a signal from outside, and that signal here is provided by your killer lymphocyte. Okay, so the killer lymphocyte will have something called as Fas ligand. Okay, so Fas ligand is present in your killer lymphocyte. So whenever this Fas ligand interacts with the Fas receptor, so Fas receptor is present in the target cell, which has to be and which has to undergo apoptosis. Okay, so Fas ligand interacts with Fas receptor. Now inside this Fas, inside the target cell, you have the death domain and FADD. Okay, Fas associated death domain. Now this will cause a death inducing silencing complex. Okay, whenever the Fas ligand interacts with Fas receptor, it activates the death inducing silencing complex, DISC. Okay, this DISC is in turn going to activate caspase 8. So caspase 8 is what kind of caspase? It's an initiator caspase. Okay, now once the caspase 8 has been activated, the caspase 3, which is an effector caspase, will be activated and the cell will be marked for apoptosis. Okay, this is what we call it as caspase dependent extrinsic pathway. The Fas ligand in the killer lymphocyte interacts with the Fas receptor and activates the death-inducing silencing complex, okay, which in turn activates the caspase 8, which is an initiator caspase, and now this caspase 8 activates caspase 3 and marks the cell for apoptosis. This is what we call it as extrinsic. Extrinsic because the signal for apoptosis is from outside. Okay, I hope it's clear. Now we shall move on to, yes, so this is a flow chart, death ligand and the death receptor. Okay, so which is a death ligand here? Fas ligand is a death ligand. This is a death receptor. Fas receptor is a death receptor. Then you have the adapters like FADD. Okay, FADD, and you have DISC formation, death inducing silencing complex formation. This is very important. In extrinsic pathway, initiator caspase is caspase 8 and the effector caspase is caspase 3. Okay. Next, caspase dependent intrinsic pathway. Okay, intrinsic pathway means the signal for apoptosis is from inside the cell. Okay, from inside the cell means here you have your mitochondria. Okay, mitochondria dependent pathway. Intrinsic pathway of apoptosis means you must remember mitochondria. So this mitochondria will have a lot of cytochrome C molecules. Okay, so this cytochrome C molecules, when a cell has to undergo apoptosis, these cytochrome C molecules are being released by the mitochondria. Okay, the mitochondria releases cytochrome C molecules. Now this mitochondria which has released cytochrome C molecule, okay, will act as an initiator for the apoptosis to occur. Okay, the cytochrome C now interacts. The cytochrome C now interacts with APAF. Okay, you can see the APAF and they form an apoptosome. Okay, they form an apoptosome, formation of apoptosome. Okay, now this apoptosome uh, actually activates your procaspase 9. Okay, now procaspase 9 will become caspase 9. Okay, then the apoptosis of the target cell will takes place. Okay, the initiator for the apoptosome formation is intrinsic. The mitochondria releases cytochrome C molecules. So those cytochrome C molecule along with APAF form apoptosome. Now this apoptosome acts as an initiator for the binding of procaspase 9, which in turn activates caspase. Okay, fine. Yes, this is again a flowchart for intrinsic pathway. Mitochondrial changes will occur. Cytochrome C will be released. Apoptosome will be formed. Cytochrome C interacts with APAF. Okay. Now that will later on lead to the activation of caspase 9. Caspase 9 again activates caspase 3. Okay. I hope I am clear. Fine. Next. Next is Granzyme pathway. Okay, so this Granzyme pathway is basically initiated by your cytotoxic T cells. Okay, so you all know there are different types of T cells: the cytotoxic T cells, helper T cells, okay, and your suppressor T cells. Still there are a lot, okay, but the basic types are these. Okay, so the cytotoxic T cell, the name itself tells they are toxic to the cells. Okay, produces something called as perforin. Okay, so perforin activates granzyme B. Perforin activates granzyme B. Now, once the granzyme B is activated, what happens is it actually releases caspase 10 and again activates caspase 3. Okay, so this is the most simplest way of putting the entire pathway. This is the main key points which you all have to understand. Okay. Now, what does this caspase 3 do? We have to understand that, right? So, if caspase 3 is a effector caspase, now once the caspase 3 has been activated, what does it do? It causes DNA fragmentation. Okay. It causes degradation of cytoskeletal structures and nuclear protein. I have already told you what it is. Okay, then it causes cross-linking of proteins, formation of apoptotic bodies, then finally expression of ligands for phagocytic cell receptor, finally uptake by phagocytic cell. Then finally what happens, it has to express certain ligands on the membrane of the apoptotic cell in order for the phagocytic cell to recognize it. Okay, so this is the effect of caspase 3. Okay, be it your extrinsic pathway, be it your intrinsic pathway, or be it your granzyme mediated pathway, this will be the final effect of activation of caspase 3. Okay. Now, there are a lot of genes which are involved in apoptosis. Okay, anti-apoptotic genes. So, what are anti-apoptotic genes? They are not going to allow apoptosis to occur. Okay, so this again is very important from exam point of view. So, BCL2 and BCLXL are two anti-apoptotic genes. So, what does this BCL2 do? It is going to control the release of cytochrome C from mitochondria. Okay, it's going to stop, or it is going to regulate the control of or regulate the release of cytochrome C. Okay, next you have your pro-apoptotic genes, which include your BAX, BAD and BIC. Okay, very important to remember. Just remember the names. These are going to promote apoptosis. So two kinds of gene, anti-apoptotic and pro-apoptotic genes. Okay. Yes, so this is all is about basic idea of apoptosis. So we have covered what is apoptosis, what are the characteristics of apoptosis, what are the different pathways involved in apoptosis, okay, as well as the genes involved in apoptosis. Okay, so do subscribe to our channel. If you have any queries, you can always mail us at our email ID, and do follow us on our Instagram page. And yes, thank you for all the support. Do not forget to subscribe this video, subscribe to our channel. Thank you so much.